So thank you very much for coming to this workshop. I'm Misha Kapushesky. I'm founder and CEO of Genestack. And let me just give you an outline of what's going to happen. So we have here Keywan Hassani-Pak from Rothamsted Research and Philip Bayer from University of Western Australia. And they are the reason why we Genestack are here. And for us, it's our first PAG. And even though the company is more than 5 years old, we started off from a very general fundamental genomics, bioinformatics, R&D infrastructure beginnings. And we got into plant genomics through a collaboration with Rothamsted Research funded by Innovate UK, which is the UK government body that funds partnerships between industry and academia that are aimed to bring industry and academia and together and lead to commercialization or knowledge transfer. Anyway, what we'll do is I'm going to pass the baton over to Keywan, who is going to tell you about the tools that have been developed at Rothamsted, and then Philip will tell us about the kind of things and kind of challenges that these tools help overcome and solve. And then we'll tell you a little bit about the company and the Genestack platform and what we were able to do. And the challenge that we faced just under a year ago was to take these tools that were developed at Rothamsted and make them more robust, make them more scalable, make them more flexible, and integrate them into a more complete genomics infrastructure. So to help people go from raw data through to primary analysis, secondary analysis, and tertiary analysis interpretation. So again, thank you very much for coming. Keywan?
Hello. Yeah. Okay. Okay. Get else. Okay. So yeah. Thank you for the introduction, Misha. So yeah, I'm very excited to talk about this project because it's really a unique opportunity for us at Rothamsted to bring the tools we are developing to a wider audience. So the project that I'm going to present here is called KnetMiner. It's developed in the last 10 years at Rothamsted Research. We are one of the research institutes in UK. We are just north of London. The crops we are working on are mostly wheat, willows, and some other crops. So let me give you just a quick introduction of why we are actually developing this sort of tool. So our research institute, like many others as well, is interested in many different traits, for example, in flowering, defense, development, stress tolerance. So, and scientists at Rothamsted design different types of experiments to identify candidate genes for this, for those traits. So we do GWAS experiments, we do different omics experiments. So, and then there is this challenge now, even if we get the outputs of these experiments, to link genotype to the phenotype. And one of the challenges really is that most of the traits that we're looking at are polygenic, complex, and it's just not really easy to understand how is this genotype leading into the phenotype. There's a lot happening in between. So what we are trying to do at Rothamsted is develop tools to build this, what we call knowledge networks. I'll talk about it in a moment, what they are, and develop algorithms and data mining tools that help biologists to link genotype to phenotype. So the tool we have started working on in beginning of 2000 is called ONDEX. It's a free and open source data integration platform that allows the integration of different types of data from different file formats. So I'm not going to spend much time talking about ONDEX. The link is here. All I can say is it has a graphical user interface, it has a Linux command line, it's got APIs to bring different data types together and create this sort of knowledge networks. So the backend is a graph data warehouse. So last year we published a paper describing how we are building these knowledge networks. examples we're given in this paper are mostly for barley and wheat and Arabidopsis. So we are talking about what data sets we are bringing together that are important for gene discovery and how we are linking it up. The approach on it's generic and can be applied to really any other species as well. So let me give you a quick recipe of how we're building this sort of knowledge network. So let's start with getting a GWAS data set. So for example, we can go to Ensemble Plants, use Biomart, and download a table like this one where you've got your SNPs on this side. On the other side, you've got the phenotypes, and then you've got a p-value that tells you how strong the association was. And Ensemble allows you also to extract the genes that are in proximity of this SNPs. So this is how the data looks like in a table form. And the problem with tables is it's really hard to understand how a SNP is connected to the phenotype. It would be much nicer to visualize it as a network. So this is the same data set now, visualized as a network. So let me tell you a bit about this, what you can see on this screen. So we've got 3 different shapes here, and they've got different meanings. So this blue triangle here is representing a gene. So this gene has 1, 2, 3, 4 SNPs in its proximity. So they can be up to, for example, 1,000 base pairs away from that gene or within that gene. And these 4 SNPs have been associated with 4 different traits. So for example, with storage, 56 days, with flowering time field, flowering time duration greenhouse, and anthocyanin 16. So they are GWAS experiments. So, okay, so here we looked at one gene. So if we bring another gene into the picture, then what we can see now is a connection suddenly. So for this particular trait, storage, we can see that there is another association. So this SNP is also associated to this trait. And SPA4 is another gene that is in close proximity to this gene. Okay, so we've now, let's say, integrated it. We just brought a GWAS data set and translated it into a network. What we'd like to understand now is, so these 2 genes are somehow both related via their SNPs to this trait. So could it be that on cellular level, those 2 genes or their gene products are actually interacting? So let's go to another database called BioGrid and download protein-protein interaction data for Arabidopsis in this case. So BioGrid has about 35,000 interactions that we can download. And what we can do now is just integrate it on top of this network. So we are overlaying now protein-protein interaction data on the GWAS genomics data. And what you can see then is that suddenly SPA4 and this other gene, ELF3, become connected through protein physics. So this is protein-protein interaction data from BioGrid. So via indirect connections through these other genes, they are now connected. So I think hopefully this is clear how the idea is clear how we just take in raw data, we choose which data sets we want to bring together for our applications, and then create networks. Obviously, we need to bring much more data together to be able to make correlations between genes and phenotypes. So for example, we can go and get gene-go relationships from other databases, or gene-phenotype relationships, gene publication, gene pathways. So we can use the same principles to generate networks. And what we can also do, and scientists actually doing, is also looking into homology, using homology data, for example, to yeast or to other crops, and bring that sort of information into the networks as well. So what we get at the end is a huge network with more than 500,000 nodes, with more than 1 million relationships between those nodes. So let me give you just a quick example now of how a knowledge network can look like for a crop. For example, let's look at wheat. So these are 3 homologs in wheat, 5A, 5B, and 5D. And these 3 homologs, they are encoding these 3 proteins here. These 3 proteins have an ortholog in Arabidopsis called TTG2. And ortholog in Arabidopsis now has all this wealth of knowledge. For example, it has protein-protein interaction data. It's got associations to phenotypes. It's got links to GO terms. It's got links to publications. What we also do is we establish through text mining some links that were maybe not in structured public databases. I talk about the text mining a bit later. So, okay, so this sort of network can be very, very large. So, okay, so what was the next thing we had to do? So now we have built these massive networks, and 3-4 years ago we said, okay, so it's nice we've got the networks now, but we don't really have the tools to mine and visualize those networks. Especially, I mean, we want to create tools that biologists can use in a user-friendly manner to relate their data to the phenotypic data sets. So we had to come up with methods to evaluate millions of relationships in these knowledge networks very fast, prioritize candidate genes, extract subnetworks, and then present that to the biologists so they can actually use their domain expertise and their knowledge and apply it to study certain traits and phenotypes. Because interpretation should be really the task of biologists. So the tool we've then started developing and still working on actively is called KnetMiner. It's free and it's available at this URL here. And what I'm going to say is just a quick word about how the bioinformatics infrastructure works. So we've got a server which is holding these big networks in memory, and we've got an API and so to query this database, extract parts of it, and visualize it on any could be desktop PC, could be mobile devices. So you'll see in a moment how it works. So okay, so the main questions I'm going to really focus on now is, once you've done your GWAS or your RNA-seq experiment, which associations genes are worth following up? So this is quite often a very subjective question, and biologists make very subjective decisions often without evaluating all the knowledge about candidate genes. And how can we just, how can we link genotype to phenotype? So let me give you an example. What I'll do is I switch to, we do that live now. Say, how can we go to the desktop? Here we go. Okay, so this is the KnetMiner landing page. You've got a link to GitHub, user guides, and publications there. You've got introduction videos, and we've developed KnetMiner instances for a couple of plant and crop species. So let's go to the Arabidopsis one. The user interface looks very simple. So it's a bit like a Google-like user interface. So what you can do is you can start typing a query. For example, I'm searching for dormancy here. And while you're typing, the system is giving you real-time messages. It's telling you dormancy is matching 216 documents in the knowledge graph, and those documents are linked through graph traversal to 8,000 genes. Sometimes biologists don't know really what queries to use, so we've developed a little query suggester wizard that can help you to narrow down your search terms. For example, you can say, I mean seed dormancy or tiller bud dormancy, so you can replace this main first query with a more specific query. Okay, so what I like to do now is we've got 3 example queries here, so we are going to focus on example 1. So I'm going to search now for flowering FLC, and the reason I'm doing that is, maybe I should have said that, is so I like to focus on a particular example. So we're going to look at GWAS data. So in this paper, Atwell et al., they published 107 different GWAS studies and looking at flowering, defense, ionomics, and developmental traits. So quite often in GWAS, I mean, this is the nice GWAS outputs where you've got the sharp peaks and they are you've got very strong associations with candidate genes, but quite often you've got this sort of GWAS outputs where the SNPs are all over the place and the peaks are the mountains, and it's not really clear to find the biological story how those SNPs are linked to the phenotype. So I'm going to focus now on this particular GWAS study, which is looking at flowering locus C gene expression, and you'll see that there are lots of peaks in here, but it's quite diffuse, it's all over the place. So we want to identify the candidate genes that are maybe most likely to be related to that trait. So now back to the demo. So the query terms I'm using here, flowering FLC. So I do a search, and so this is the first time I'm using NetMiner on this PC here. So apologies if it's sometimes not working or too slow because the internet is not very stable. So so we did a search, and what we get back in this view is a ranked list of candidate genes. So the genes are listed here. We are calculating a score for every candidate gene, and we are demonstrating the evidence types that were used in this column here. So for example, this is representing phenotypes, this symbol is representing gene ontology terms, this is representing SNPs, and so on. So there are different views to look at the data. We can look at it in a table, or we can switch over to a chromosome viewer called genomaps. So in genomaps, we can then go and so this button here makes it bigger. Hopefully you can see it at the back. So we've got now on the left-hand side, we've got the SNPs from the GWAS study that are associated with FLC, and on the right-hand side, we've got the candidate genes predicted by NetMiner. What we can do is say, show me just the top 100 ranked candidate genes from NetMiner, and we can filter the p-value to look at less SNPs, for example, in this view. And now we can go and zoom into the regions and select certain candidate genes that are overlapping with candidate SNPs. So for example, here's a little area where SBA4 is overlapping with a gene, so I can select this one. And let me select just a couple of more to show you that it's not always just selecting one gene and looking at the story. We can select multiple genes and then press this button here. So now something more complex is happening in the back end. Is it's going and extracting for these 4 genes that we have selected in that view, the network, and it's visualizing you the network for these 4 genes to show you, are they maybe connected through pathways, through protein-protein interaction data, through co-expression data, and what other knowledge is known about those genes. So that's a task that would take a biologist a long time to go. It's a very technically demanding task to go get all that information and visualize it in a nice way. So here we get it quite quickly, and biologists can actually now spend time to interpret the data rather than spending time on the technical challenges. So let me give you an introduction now to this network viewer. So I'll make that big again. So every node has a different meaning here. So I introduce you to the different nodes. So this is the green rectangles represent phenotypes. If you click on this one, you'll see there is a description, for example, late flowering, flowers later than FLC3, single mutant, and so on. Then this is representing a gene ontology term called control of flowering. Triangles represent the GWAS study. The description is given here. Stars represent SNPs. And relationships between entities can have values attached to them. For example, the SNP linked to that trait has a p-value attached to it. So if you click on it, you can see the p-value shown here. Okay, so what we can do now is let's go and, for example, filter certain types of information. So we don't want to see the publications that are in this network. So you see the publications that are linked to certain genes that are shown here. The keywords are highlighted, but for now I'm going to hide certain types of information. So I'm hiding publications, I'm hiding phenotypic data now, and just focusing on this subset. Okay, so what I can do now is add labels to the genes, for example. So these are the genes. So you'll see FLC was one of the genes we have selected, SBA4 was the other one, they've got a double border, LD was one, and FRI was one. So now we can see, wow, they're all part of a network, and the black relationships represent protein-protein interaction data, green relationships represent gene ontology annotations. So I'm adding now labels to the gene ontology annotations and finding out that those 4 genes seem to be involved in vernalization response and regulation of photo and so on. Okay, so I'll stop here. Just want to give you, there is a lot of story that in those networks, and a domain expert in biology can read those networks easily and generate new hypotheses. So do I need to click on this one, Kevin, or doesn't it? Okay, second use case I'm going to just spend a bit of time on it is an RNA-seq experiment that we've done at Rothamsted to understand really sprouting and grain color relationships. So pre-harvest sprouting, let's say, is just a negative trait that we don't want to have in our crops, and grain color, we can have white grains and red grains, and white grains are more prone to pre-harvest sprouting. So an RNA-seq experiment was designed to look at the differences on transcriptomics level between white and red grain, and the red dots are differentially expressed candidate genes. So it's still hundreds of them. So really the question Andy Phillips, who was working on this project, for example, was asking is, so how are these 100 genes linked to grain color and PHS? So what we can do now is go to NetMiner, write down some knowledge that we've got, for example, about the traits we're looking at. So we know pre-harvest sprouting, related to dormancy, so we enter dormancy there. We can use Boolean expressions, say or color, or flavonoids, or proanthocyanidine, we can type that here and enter the list of candidate genes in this box and press the search button again. So let's do that live again, so you really trust and believe me that it's working, and I'm not just talking here. So to do that, we need to go to a different version of NetMiner. So now we are in wheat. So let's jump to the wheat version, which is here. So the wheat NetMiner is currently based on TGAC version 1 gene annotations. And the example we are going to use is example 3, grain color and PHS. So I'm entering the keywords at the top, candidate genes in this gene list box, and pressing the search button. So obviously it's lots of genes and the networks are big, so it's doing all the calculation now again to extract the knowledge networks for these genes and find out if any of these genes in this list could be maybe related to any of the keywords at the top. So that takes a while, but then you're presented with this view called the gene view again. We can sort the table based on this user column to look just at the genes that were entered in this box. So what I'm doing now is just one click. I'm selecting all the candidate genes that were entered in the box here and saying, show me the network. So again, it's going now to, for all these genes, extract the network and show us if they are related, show common pathways, interaction data, and so on. So once we get the networks, it might take a while, but it's here now. I'll just show you one piece of information that I didn't talk about, is the text mining. So, okay, let me zoom in a bit and add labels to this. So what you see here now, this green pentagons are representing trait ontology terms, and the blue relationships, they are automatically generated text mining relationships. So we can now go and say, why was this gene linked to this trait ontology term? We can click on this relationship, and what the system has done is it has extracted from all the publications that are in PubMed the evidence sentences that have linked the gene to that trait ontology term, and it's presenting the biologists just that sentence, so they don't need to read the whole paper. And obviously, text mining has lots of false positives. So sometimes they might trust it, sometimes they might not trust that information, but it's them making the decisions at this stage. Okay, so left with a couple of summary slides. So anyone interested to know how the text mining works? It's published here, but we use a very simple text analysis methods to link our concepts to publications, and then create co-occurrence networks between things that were linked to the same publication. Okay, so the methodology is published here, and we can discuss it after this workshop, maybe if you're interested to know how the text mining works. How does the gene ranking work? So gene ranking works using TF-IDF techniques to find the documents that link to the keywords. And then we use some network analysis techniques and information theory to identify which concepts are specific to the genes or are linked to lots of genes, then we don't weight it so high. So the most important thing for us is to come up with a scoring method that is really fast. So we want to have within seconds, we want to compute a score for thousands of genes. So that was very important. So, okay, so just give a summary of NetMiner. So far we had the pleasure to collaborate with lots of smart people like Philip, Bea, and people from IPK and people in Argentina at Inter to develop NetMiner for different species. Some of them you can see here. So it's still quite a bit of work involved creating new NetMiner instances. So you need to know how to build those knowledge networks, set up your own servers. So that's why we are collaborating with Genestack to make that just easier and develop an approach where everybody can develop a NetMiner for any species. Okay, so people at Rothamsted I like to thank are especially Ajit Singh, who's a software developer in my team. He's done a lot of the visualization. We just were successful to get a funding to develop a new version of NetMiner called Disease NetMiner, and the aim there is to link up cross-kingdom, create a cross-kingdom NetMiner which links the crop pathogens to the crops really. And Marco Brandisi is working on that project. Chris Rawlings is here as well. He's head of our department and was behind the Ondex project. So, and all these collaborators here contributed to the project. Thank you to everybody, and thank you for listening.
I think we have some time for a couple questions.
Sure. Yeah, happy to take any questions if there are any questions, or we can take them.
Yeah, I've got a question.
Yeah.
So it's a really nice tool, but I mean, it's only as good as the data on which it is based, right? So if I publish a paper tomorrow about flowering time, when am I going to see it on your networks and your software?
Yep. So this is something we've been working on last year to automate the whole network generation process. So I would say we've got it now 80 to 90% automated. So what you see here is there are now scripts that go and download PubMed, download all the different databases, and then run Ondex in the background to generate the networks deployed on a server. So once it's fully automated, you can just set up a cron job that does it every month or every 2 months. So right now we're trying to update maybe with every ensemble release, we try to push a new NetMiner version as well for every 2-3 months roughly. But there's no reason once it's automated why you can't do it more frequently.
So what about PubMed, for example? How often is that updated with your software?
Yeah, so we don't use the whole of PubMed, right? So for example, for the Arabidopsis knowledge network, we are just downloading the subset of PubMed that mentions the word Arabidopsis in the abstract. And that's about 60 to 70,000 publications. And it takes about half an hour to download that subset from PubMed using eUtils and eFetch. And then it takes another 10 minutes to generate a network out of that subset and integrate it with everything else. The whole process takes maybe 7 to or maybe less now, 2 to 3 hours download, generate a new network. So we can do, yeah,
Thank you.
often if you want to.
I mean, the supplementary point is that if you've got private data that you want to have incorporated in the network, then there are mechanisms to make that, and you're in control of that entirely. And that's also part of the motivation for the work of Genestack, where you're wanting to mix private information that you're in control of with the public domain stuff